This procedure aims to create objects and object categories for studying how we perceive and learn to perceive objects by sight and/or touch. First, virtual morphogenesis, or VM, is used to simulate the processes of early embryonic development and create novel, naturalistic virtual 3D objects called digital embryos. Then, using virtual phylogenesis, or VP, object categories are created with precisely defined statistical properties based on the input digital embryo. If desired, principal components analysis can be used to create additional shape variations among the virtual objects created by virtual morphogenesis and virtual phylogenesis. The likelihood that a given object belongs to a given category can be precisely calculated using feature-based Bayesian inference. If necessary, haptic prints of the resulting virtual objects can be generated using a 3D printer. Each of these methods will be illustrated in greater detail later.
Compared to existing methods, these new approaches create naturalistic but precisely measurable shape variations that arise without the need for investigator-imposed constraints. They offer novel tools in the fields of visual and haptic perception, perceptual learning, and machine vision, and have potential applications in the rehabilitation of many types of visual impairments through visual haptic cross-modal training. Interestingly, this method can also be applied to study the processes of morphogenesis and evolution themselves.
We first had the idea for this method when we were looking for ways to generate naturalistic but precisely definable visual stimuli for studies in computational vision. Initially